Nicholas, Minnesota, a small town about 2 hours north of the Twin Cities. And although he was surrounded by his mother's beautiful garden from a young age, his interest in plants and plant pathology began in college at the University of Minnesota. In summer, he worked in 2 conservation core programs in Vermont and Colorado when he participated in ecological restorations and trail maintenance projects in the Forest Service. He changed his major to plant science and graduated from CFANS in 2016. Later, he worked as a gardener and as a researcher in Eugene's lab at the USDA Cereal Disease Lab. And in January 2019, he began work as a graduate student. Nick has excelled academically. He maintained a 4.0 GPA, and his work is funded by a grant through MITPPC that he authored for the first year of graduate school. Nick has already 1 manuscript and 2 first reports that are in press in the journal Plant Disease, and there's a 2nd manuscript that has already been accepted for publication. He has taken many opportunities to share his research, and he presented it numerous times at APS, the Upper Midwest Invasive Species Conference, the North American Rust Workshop, and at the Site Pride events. In our lab, in addition to his own research, he has contributed substantially to various other projects, including the phenotyping of foreign stem samples and surveys of hybrid barberries. And honestly, it's been great having Nick as a graduate student in our lab. We have learned a lot from him. He has been an excellent student, and I think you will all enjoy a great summary of what has been his great job as a student. All
right, thank you very much. And thank you all for coming to support me in the final step of my PhD. I titled my presentation, Studies on Puccinia coronata, var coronata, and other recently observed rust fungi in Minnesota. And as you can probably guess, let's see here. There we are. The rust fungus that you see on screen is this Puccinia coronata, var coronata. And this is interesting because 10 years ago, you wouldn't have seen this around Minnesota. This is a new disease that has emerged, and it's likely a new introduction to North America and to Minnesota. And Eugene noticed this for the first time in 2017 or so. And shortly after, greenhouse inoculations and field observations showed that the grass host of this fungus is reed canarygrass. And reed canarygrass and glossy buckthorn in Minnesota grow right next to each other in wetlands very densely and create just the perfect environment for this fungus. And here they are growing right next to each other. And if you go to a wetland around the Twin Cities in a month or two, you can see glossy buckthorns that might look something like this, with significant defoliation and flower and fruit loss due to rust infection. And on the right, we have a picture of glossy buckthorn with all the leaves pulled away, so you can see the flowers that are actually infected by the PCF. They're actually supposed to be white, not this orange color. And you can even find them kind of beautiful and even suitable for a boutonniere. Personal taste. Yeah, and they're really interesting fungi. And you can also see a significant infection on reed canarygrass, causing premature leaf senescence and defoliation, presumably with some effect on plant fitness and competitive ability. So noticing that we had a likely introduced fungus in Minnesota, that is a crown rust fungus, very closely related to major pathogens of oats and turf grasses and barley, and that it's affecting 2 highly invasive species, glossy buckthorn and reed canarygrass. We put together a grant to the Minnesota Invasive Terrestrial Pests or Plants and Pests Center with 3 primary goals in mind. First, to look at the distribution and identify it and do some phylogenetics work and improve the description of the pathogen. Second, to look at the host specificity on grass and buckthorn species. And third, to evaluate its potential as an augmentative biological control agent of its host. And these would eventually become chapters 1, 2, and 3 of my dissertation, and they'll also structure our talk today. But it turns out that if you spend enough time just walking around outside and trying to identify the rust fungi around you and sequencing them when you have the chance, you might find some pretty interesting things. Some of those interesting things have ended up in my dissertation. So we have chapter 4, the host specificity of Puccinia digitati coronata, a new crown rust fungus of Kentucky bluegrass in North America. And I'm very excited about this paper. We just submitted it to Plant Disease. It's the first report of a new coronavirus pathogen of the most important turf grass in North America. And lastly, we have briefly two first reports of Puccinia glitchomatis, rust pathogen of creeping Charlie, and of arrestive lemongrass in Minnesota. But before we get there, I want to take a step back and talk a little bit about some background information. So first, invasive species. When I began to learn plants and trees and shrubs and grasses in college, one of the things that shocked me was actually how many of the species around us are non-native plants and invasive plants sometimes. And while it seems kind of like a trivial thing, it's really not. And invasive species globally are among the top drivers of biodiversity loss, along with climate change, habitat loss and fragmentation, and overexploitation of resources. And the transportation of species across natural boundaries, for example, through international trade across continents, is a feature of the modern world. And new things are coming in all the time, both plants and pathogens. At worst, invasive species can interact positively in what Simberloff and Van Hal call invasion of meltdown. And common buckthorn is really the textbook example of this. It's the ultimate host of the oak coronavirus pathogen, of course, like an introduced species. And it's also the overwintering site of the soybean aphid and also facilitates invasive earthworms in Minnesota. But other organisms naturalize with limited impact. And some of them, like a classical biological control agent, for example, or in our case, Puccinia coronata var coronata, seems to even counteract the effects of other invasive species. And I think this is a really interesting topic of research. Canarygrass, or Phalaris arundinacea, is a large, highly rhizomatous grass. And if you take a look at this photo and remember it, you're going to see it everywhere in about a month or so around the Twin Cities metro area. It's native across the temperate northern hemisphere, including in North America. And because it's invasive, people often mistakenly assume it's actually an introduced species, but Neil Anderson and Alan Smith and their group have really convincingly shown that it's actually a native species in North America. It's widespread and highly invasive in Minnesota, as well as other parts of North America, and it was likely spread throughout Minnesota as a forage, contributing to the propagular pressure and probably its invasivity. It's also highly advantaged by nitrogen pollution, sedimentation, and intermittent flooding, changes in hydrology. So as you can imagine, an urban wetland with lots of stormwater runoff and some yard fertilizer mixed in there is a really ideal setting for a canary grass. Glossy buckthorn is a small tree. It is native to Europe, Western Asia, and parts of North Africa. And historically, it has used as a medicine and a dye. And personally, I've experimented a little bit with dyeing with this. You can get between like a green or a gold color, as you can see in my sweater, although this was actually dyed to take common buckthorn berries, which is what I had on hand. The first American herbarium record is from 1879 in New Jersey, and since then it's been spread throughout the Midwest and Northeast as an ornamental shrub, including actually right outside my first apartment when I started working on this research project. There's like 5 huge glossy buckthorns right outside this building. And actually, when I was parked my scratched up old Honda Civic under it, after a few weeks every year, it would actually rain the easiest course onto my car. I took this as a good sign. It's a major well invasive in Minnesota, like canary grass. And now crown rust fungi. So probably all of you think you know about crown rust fungi, and you know it as a oak crown rust as a pathogen of oats, and that's true. But there's a lot more to the story. And actually, the first crown rust fungus described is Puccinia coronata var coronata, the topic of my talk today. It was originally on a Calamagrostis species, and the original holotype is now the holotype of Puccinia coronata var coronata, or as I'll call it sometimes throughout the presentation, probably PCC. Its crown rust fungi, Puccinia coronata sensulato, are now organized into Puccinia series coronata, according to the more recent phylogenetics work. And this series contains major pathogens of cereals, of course, including the oak crown rust pathogen, which according to the most recent work is kind of a mouthful, Puccinia coronata var veniformis specialis. And unfortunately, I'll have to say that a few more times this time. Thank you. And the barley crown rust pathogen, Puccinia coronata fortiae. In total, we have 17 named species with more varieties, Forma specialis, and some unresolved lineages. So we still need some work. And often these fungi are alternating with widespread invasive common buckthorn or glossy buckthorn, Rhamnus, pathetica, and Frangula, alnus. And likely owing to the widespread buckthorn hosts, we actually have a couple of recent emergences of various crown rust fungi in North America. And this can be due to acquisition of virulence on grass hosts maybe, or due to introductions of new pathogens in North America. So we have in 1991, the occurrence of crown rust on barley. And this is work done by Eugene and Brian Stephenson in the 90s. We have in 1995, brome crown rust appearing on smooth brome, common forage and widespread invasive grass in the Midwest. This rust is absolutely everywhere in the Twin Cities area. In the 2000s, we have the emergence of Kentucky bluegrass crown rust in the Puccinia coronata variviniform specialis and beanie clade. In 2011, we have a first report of Puccinia coronata varigiporosa, although this one is probably not widespread apparently, on blue oatgrass. And with my work, we have canarygrass crown rust, PCC, also written up in a first report out in Cornell by Kennelly et al. And now a second Kentucky bluegrass crown rust, Puccinia digitata coronata. And briefly, just to look at rust fungi as biological control agents, rusts are very highly valued as classical biological control agents. So that means going to like an invasive organism's original range and finding pathogens or pests of that species, bringing them over and letting them loose, not doing too much more than that. Rusts are highly host specific and they're prolific, often producing a lot of spores that spread a long way. They do a lot of the work for you. And of 36 fungi approved as classical biological controls, actually worldwide, 24 of them are rust fungi, and including 4 in the United States. But there have only been 2 examples of augmentative biological control agents, what we're talking about today. And these are also called bioherbicides. And neither of them, unfortunately, were successfully commercialized. Despite interest and despite good proof of concept research, due to difficulty in commercialization, yet we don't have successful examples currently. Long-term success requires secure funding. This doesn't mean that you can't do it, but it's just something you have to consider in coming up with a product. And that brings us to the main body of research. And unfortunately, yeah, there are 5 chapters here, so we're going to have to go kind of fast and gloss over some interesting details here. Yeah, find me after, read my dissertation if you want, or the publications preferably as they come out. So first, we have Puccinia coronata var coronata, a crown rust pathogen of 2 highly invasive species that's detected across the Midwest and Northeastern United States. And this was published in Plant Disease, or it's in press and available online as a first look. Nicholas Greatens and Leslie about the Serial Disease Lab are co-authors on this, along with Pablo and Eugene. So our research goals here are to improve the description of PCC, since the most recent one was just on using teliospores, to determine the identity of samples with a phylogenetic approach, and to assess the distribution of PCC in Minnesota and in the United States. Broadly, the methods are surveys throughout Minnesota in 2020, and to actually solicit samples from users of iNaturalist.org from across the United States. I didn't anticipate this at the beginning of the work, but because this is apparently so widespread and on a widespread and widely loathed invasive plant throughout highly populated regions of the United States, people are taking pictures of this all the time and posting them online. And really just by being on iNaturalist and messaging them, I was able to significantly expand the number of states where we could have reports of this pathogen. And then lastly, we sequenced amplicons of 2 to 4 loci per sample, beta tubulin, ITS, RPB2, and COI loci, following recent phylogenetics work. And we actually used a nanopore. Usually we think of nanopore as more for genomics purposes, and it's like long reads that are useful in assemblies, but it's actually very good in amplicon work as well. You can get longer PCR products to sequence than you can do with Sanger sequencing. And if you maximize your efficiency, you can really get extremely low cost per amplicon. We didn't do this in this project, we will in the next, but yeah, less than like 10 to 25 cents a sample even. And this is likely to replace a lot of Sanger sequencing probably in the next few years or decade or so. Oops. So this is the first figure from our publication. This is macroscopic science Puccinia coronata var coronata, and this is part of the description part of the paper. So in A, we have the spermatozoa or Puccinia. These are going to be fertilized and produce ECI, causing loss of flower and fruit production and big large growths. On D, we have the buffer a month after infection, and then E and F for production of arundinacea. And we also have a figure for microscopic spores and spore structures. And one thing I like about this paper is we actually are able to see all 5 spore structures that you'll see in macroscopic or macrocyclic rust fungus. This is kind of a rarity in rust work just because these stages are kind of ephemeral. But in this case, we have images of the Puccinia here with the hyphae, the fertile hyphae extended out to catch compatible Puccinia spores in B. And then C and D, we have EC spores and arundinacea spores. All the SEM images are from Dr. Gail Celio at the University Imaging Centers. E teliospores, and F, we have a teliospore that's actually germinating to produce that promycelium, and actually a germinating spore. Our sequencing work strongly shows that the samples that we see around here are consistent with PCC in Europe, and our samples are the 20 MN and so on. Strong in European certified PCC specimens. Here we have a beta tubulin tree. This is the maximum likelihood tree, and ITS in B. And you can see the pretty strong bootstrap support, 90 in A and 84 in B. And we've also confirmed the distribution across the United States using these samples identified by phylogenetic analysis or West. So in the orange diamonds here, we have confirmed reports. And the green circles are reports of glossy buckthorn, purple triangles, canarygrass reports. And as you can see, PCC is widespread from eastern North Dakota all the way over to New Hampshire. And PCC is reported for the first time in 11 states. Samples from 70 states are from my naturalist users. And just a little Easter egg here, this sample is actually from the Borlaug's childhood farm. People are making fun of me for collecting samples on our little vacation to the World Food Prize, but that's our only report of the rust in Iowa. In Minnesota, I also did some surveys, and we find that PCC is widespread in central Minnesota and in the so the orange squares and orange triangles are reports of rust on glossy buckthorn and canarygrass. Haven't confirmed these ones as sequence. But you can see that lots of reports here in the central part of the state, but when you go out into the western part or the northern part, there's actually no rust on the canarygrass. And this is late in the season when you actually expect it to be there. And I don't know why this is. It is the case that these hosts are susceptible. I brought them back and tested them in the greenhouses, and sure enough, they're susceptible. But for whatever reason, this rust fungus doesn't apparently like to go very far away from its hosts. And this is kind of contrary to what we'd think with cereal rust fungi being very highly mobile and spreading, across a continent even. That's not the case here. I don't know why. So in short, local samples are consistent with PCC since we in Hamilton. The most recent phylogenetics work. It's widespread across the Midwest and Northeastern United States on glossy buckthorn and canarygrass. And we improve the description and show how community science can be useful to map the occurrence of a new and introduced pathogen. And we'll just move right along to chapter 2, looking at the ecelial and cilial host specificity of Puccinia coronata by coronata. This manuscript was accepted in Plant Disease, and Eugene and Pablo are co-authors. Our research goals are to determine the susceptibility of grasses and buckthorns to PCC, to assess the threat posed by a recent pathogen introduction, see if turf grasses or cereals or native plants that we care about are affected by this, like the new introduction. And if we're going to use this as a biological control agent, we want to produce a host range assessment. So in brief, our methods will inoculate potential ecelial hosts with PCC inoculum on overwintered canarygrass. And we'll inoculate potential grass hosts with bulk inoculum derived from Ecia and confirm that with pure isolates that we developed in the previous chapter. This was a large experiment. In total, we looked at 1,800 accessions or so of 6 hero crop species. And of other grasses, we looked at 44 genera, 110 species, and 783 good accessions. And 16 species of Rhamnaceae and Elaeagnaceae, buckthorn and oleaster families, which contain some species susceptible to some crown rust fungi. And this is the large composite image that was produced from the paper. And we'll go through this one by one. So first on the cereal crops, looking at oats, barley, rye, red wheat, durum wheat, and triticale, everything is resistant. The range of responses are given on a scale of 0 to 4. Anything above 2, or anything of 3 or above is species. So bread wheat, durum wheat, triticale, entirely resistant, giving us a little bit of flecking sometimes. But we are able to see some amount of sporulation on barley, rye, and oats. With some exceptions with limited successful sporulation, or what we might call a near non-host. And here we have a range of responses in oats to PCC. On the left, completely resistant. This is what we mostly see. But 2, a little bit of flecking, 3 up to like a 2. So still a strong resistance response, but some amount of sporulation. In total, we see 33 grass species actually in 5 tribes: Brachypodi, Bromine, Melissae, Poli, and Trimsea, hosting PCC with some amount of sporulation. All the susceptible species are in Poli, just like Greek canary grass. 13 grass species in 5 genera are susceptible, including Phalaris. Here's a range of symptoms on Greek canary grass, ranging from complete resistance, which we can see outside, to susceptibility. We can also see some amount of sporulation in other Phalaris species, as you might guess, ranging from complete resistance in like Phalaris canariensis, widely used for birdseed, all the way to high susceptibility in Phalaris periapopsis, introduced species invasive in parts of the state. We see some amount of sporulation on Calamagrostis species. These are common wetland species in North America, temperate regions around the world. These 2 native species are both sometimes affected. Calamagrostis canadensis is generally resistant, but even in the Twin Cities, we see a little bit of infection on this. Some Calamagrostis species, including Calamagrostis purpurascens, are of some concern in some parts of the country. And also of interest, we have these 3 grass genera, April, Lamarckia, and Puccinia. April and Lamarckia are Eurasian genera that have some species of which have been introduced in North America, and there's some native Puccinia introduced as well. I just want to point this out here because we'll talk about it a little bit later. And we see some wild oats also weakly affected. And notably, we see also Brachypodium able to be infected by this fungus. So this is the first time, to my knowledge, that we have Brachypodium distichion, a mottled grass species, affected by a rust within Puccinia coronata, sensu stricto. We tested some 52 accessions of Brachypodium distichion and a couple of some other species and hybrids, seeing a maximum of 0 to 2. And we saw this in 10 of the 52 accessions that we tested. Again, of some possible use in non-host resistance studies or something that expert if someone wants to follow up on that. Looking at the eastern host specificity now, we see, of course, Glossy buckthorn, Frangula alnus being infected, and also 3 native North American buckthorn species. Greatens lanceolata from the central part of the United States, south of Iowa or so, and then Frangula caroliniana and California from the southeast and west coast, respectively. Common buckthorn and our North American or our Minnesotan native buckthorn are both resistant. So in conclusion, cereal crops are resistant. PCCs weakly infects oats, barley, rye, and mottled grass Brachypodium distichion. 13 grass species and 5 genera are susceptible, and there's some possible conservation concern for some species. Chapter 3. We have the effects of the crownless pathogen PCC on invasive glossy buckthorn and canary grass in greenhouse trials. Our research goals here were, as the title suggests, assess the effects of PCC in greenhouse trials, and also secondarily to evaluate the potential as an augmentative biological control agent of its hosts. So we see some strong effects in natural situations where these 2 hosts are present, but we want to see if we can quantify those effects in greenhouse trials. So briefly, looking at the effects on Greek canary grass. In June, we took 80 of those Greek canary grass rhizomes that were collected from around the state as part of Chapter 1. Let them grow up in the greenhouse and inoculated half of them at a 2 to 3 leaf stage with ECS spores from glossy buckthorn. We mistreat the plants once a week to simulate a natural reinfection process. These are the arundinacea spores, so this is a natural, a polycyclic stage of the life cycle, right? And then in October, after 14 weeks, we'll assess height and tiller production of each group and harvest the biomass. And in November, as a way to look at the below ground biomass, we'll allow the plants to regenerate, and in December, we harvest the biomass. And what we see is that the plants that are treated with the rust fungus, fairly predictably, are pretty strongly affected, looking pretty with unhealthy leaves and just in poor health compared to the control group. And these were 4 randomly selected plants from the treatments. And here we have 3 randomly selected plants from each treatment. And here the control group clearly has just much more substantial below ground biomass production. And the data bears this out as well. So if we look at the number of tillers after 3 months on the y-axis and compare the treated and control groups, the treated in rust green 4 color, we have the treated group giving us about 71% control. And similarly, if we look at dry biomass after 3 months, the treated group has about 75% control. And if after we allow it to regrow from the below ground biomass, we see a reduction in height and 85% of the control, and most substantially, as you might guess, in biomass, where the treated plants are actually only producing 48% of the above ground biomass after they're allowed to regrow. So this is pretty substantial, and I think indicative that it would probably have an effect in natural circumstances as well. And next we look at glossy buckthorn. So usually I'm looking at adult plants in a natural ecosystem. I'm looking at the defoliation, the loss in flower and fruit production. If we were ever to use this as a biological control agent, we figured it might be useful in addition to knocking back fruit production to kill the seedlings. So this is one thing that we looked at. Just briefly in the methods again, collect the berries and stratify the seeds in fall. And in spring, take 100 of those seeds by weight and put them in 16 pots with 8 pots per treatment. Collect some fresh Telea inoculum from the wetland nearby. And after 3 weeks, 3 weeks after germination, we'll inoculate these plants. And 2 months later, assess biomass, height, leaf area, and I was done digitally using this nice tool using leaf area. And what we see is that we have a slight reduction in height in treated plants and a substantial reduction in leaf area. So when you inoculate baby glossy buckthorn plants when they're really young, you can really knock back the leaf production. And similarly, we can see this an effect on the biomass on average per plant. And we're seeing about 81% of the control in treated plants. We also see 6.8% mortality, and as you can see in the image on the right. So sometimes the glossy buckthorn is just killed entirely by the rust if you do it. But just 6.8% of the time, so on. So some practical considerations here. Arundinacea spores of blue canarygrass are easily collected. You can grow them up in the greenhouse or just vacuum them from outside on ample canarygrass. The ideal use would probably be early in the season, even when the spores are not there naturally, like right now or a few weeks ago, and probably in high value level for naturally limited rust. Blue canarygrass is easily hayed and could also be infected with the rust and spread onto glossy buckthorn as well. But there are some challenges in storage and maintaining viability, and because this is the monocyclic stage of the rust life cycle, it would require repeated applications. That's not to say that it's impossible, but it would just require a little bit of scientific labor to make this work, and definitely a dedicated team of people to make this happen on a regular basis. There are some concerns with non-target species, as we talked about in the previous chapter, but really, I think there's probably limited risk considering this pathogen is already very highly established, and if some these species of concern are present in your local area, you could not use the product, of course. So in conclusion, blue canarygrass biomass production is significantly reduced with rust infection. Glossy buckthorn likewise is significantly affected by rust infection. And it offers some promise for control of blue canarygrass and perhaps glossy buckthorn, but field trials are certainly needed. And we definitely want to learn from previous rust bioherbicides that were not successful. So that takes us to chapter 4, which is on a different organism entirely. The host specificity of Puccinia digitati coronata, a new groundnut fungus of Kentucky bluegrass present in North America. And this was recently submitted to Plant Disease. And thank you to my co-authors, Carl Miller, who started work on this project, research in our lab, Eric Watkins over in turfgrass and horticulture, and Eugene and Popple, of course. So a little bit more background information on this project in particular. Ground rust emerged as a disease of Kentucky bluegrass around the 2000s, according to this Barron et al. Paper. But according to the recent phylogenetics work, this pathogen is actually in the Puccinia coronata var uniform clade. And this clade, in addition to including the oat crown rust pathogen, also includes the crown rust pathogens of ryegrass and tall fescue. So the original idea for the project that Carl started on was to compare the host specificities of these pathogens with the idea that maybe if that POA crown rust pathogen also affects lolium, the ryegrass or the tall fescue, that has some epidemiological significance since these turf grasses are all growing together all the time in our yards. But then we have a description of this fungus actually just in 2022 by G et al. Over in working in Northeast China. And their ITS sequences that they recorded were near identical to some of ours that I had got from 3 East Asian buckthorn species over in North Dakota, Rhamnus granata, Rhamnus japonica, and Rhamnus duburica. So noticing that this pathogen is also in the United States now, we sequenced the rest that Carl had been working on and had already been developing host specificity data for. And turns out it's actually this Puccinia digitata coronata. So this ends up being a much more interesting research project, of course, although someone should still follow up on that other idea. Our research goals here are to determine the placement within the crown rust fungi complex and to assess the host specificity on grasses and buckthorns, just like the previous one. And our methods are to sequence 4 loci and compare with the related fungi and do a slightly smaller assay with the serial crop species, look at 383 extensions. And of the wild grasses, look at 27 grass genera, 63 grass species, and a total of 235 extensions from the grid. And here we have a big figure for you. I'll talk you through it. This is the maximum likelihood tree of various crown rust fungi inferred from ITS2 sequences. And a couple things to note here. We have everything that goes on to POA is highlighted in green here. And the crown rust fungi, Puccinia series coronata, are on top, as you can see in the array, and the outgroup taxa on the bottom. So what we see is that the samples on POA are dripping into 2 distinct strongly supported plates here. On top, we have Puccinia coronata var including all the samples from that Barron et al. Paper and the Lewin Hamilton paper, the other most substantial phylogenetics work. And then the second group here, we have all of our samples, which are the PUR ones that are submitted to Purdue or Varian, as well as a couple from another paper that just identified samples to Puccinia coronata in the broad sense without trying to identify some or through another taxon. And also we have the type specimens, the paper from China. And here we have a consensus tree from a partition Bayesian analysis of 4 loci. And here we have our samples in this case with very strong support to the Puccinia coronata census stricto, which are on top here. And there are some fungi that are omitted here. There's fewer names on the right, just because there was limited public sequencing data. So this clearly shows us that we have a completely new pathogen that hasn't, or in North America that hadn't previously reported. In addition to being genetically distinct, it's also morphologically distinct, and it's distinguished by its small uridine spores. And on the right here, we have a comparison of the crown rust pathogen in the same place as the other POA crown rust pathogen and the Puccinia digitata coronata. And you can see here that the uridine spore in B is much smaller, although there's some variation in uridine spores in a single sample. We also have very ornate teleal appendages, as you can see. These are the little tentacles that are on top of the top cell of the crown rust teleospore. And this is what gives it the name crown rust as well. And here again, some nice scanning electron micrographs from the University of Michigan Centers. On the left here, we have a side view of a teleum. And you can see this is the top cell of the teleospore with really long teleal appendages, actually up to 20 microns long, which is longer than the uridine spores themselves. And on the right, we have all of those teleospores. This is a top view looking down at the teleum, and we have all of those teleospores with their appendages interlocking. I've never seen images like this before. And again, we have a huge composite figure that we'll walk through one by one. Looking at the cereal crop species, red wheat, barley, durum wheat, oats, rye, and triticale are all resistant, although we see a range of 0 to like a FLEC 1 response in oats and rye. And again, this is on a rating system of 0 to 4, with anything less than 3 being resistant. Here's a range of oat responses to this Puccinia digitati coronata, ranging from complete resistance up to like a FLEC 1 response with some necrosis. In rye, we see similarly mostly resistance responses on the left, up to like a FLEC 1 with a little bit of sporulation again. And interestingly, some wild oat species are actually susceptible, although there's a range of responses. In Avena barbata, we see a range from like a FLEC 1 response, again, all the way up to like a 3. And in longiblumus, complete resistance up to like 2+ or so. POA species are broadly affected by this pathogen. So of the 12 that we looked at, 11 of them were susceptible, and only on the left here, POA annua, sometimes a turf grass and a common weed around here, is resistant. And all these other POA species, some of which are native, some of which are introduced, are all susceptible to this pathogen. Kentucky bluegrass is a little bit variable in response. We see some resistance in some accessions, but overwhelmingly in greenhouse assays, Kentucky bluegrass is susceptible to this pathogen. Although notably, you can see some differences in general just between field responses and greenhouse responses, of course. And in the turfgrass research plots where Kentucky bluegrass is mowed and otherwise kept in generally good health, we see less infection. Outside, we see the most infection in unmowed areas in the shade, where the environmental conditions are much better for rust infection. And we also have several of these other species susceptible again. So that's Aethrola and Puccinia species. These were also susceptible to Puccinia coronata, var coronata, right? And one interesting thing, and this actually didn't even make any specific, but so noticing that these are susceptible to 2 different crown rust fungi, we actually also tested them for susceptibility to oat crown rust, and they're susceptible to oat crown rust. And we just tested them for susceptibility to oat stem rust, and they're also susceptible to oat. I don't know why. It would be certainly the topic of a good topic for a study, but certainly a curiosity. There's some grass species that are susceptible to a diverse array of rust fungi. Brachypodium gives us a response of nothing to a little small bit of necrotic flecking. And now the East host specificity, again, we have those 3 East Asian buckthorn species that are all susceptible under natural circumstances. And also we have Rhamnus separatica, common buckthorn, and our North American Rhamnus lanceolata also susceptible. So what this means is we have a widespread invasive shrub, Rhamnus separatica, common buckthorn, and a widespread turf grass, Kentucky bluegrass, and invasive species, sometimes Kentucky bluegrass. 2 of the most abundant plants in Minnesota are the 2 hosts of this new introduced pathogen, Puccinia digitati coronata. And probably in some of your yards and on campus and all over the place, this life cycle is happening. So in conclusion, we have 2 distinct crown rust fungi found in Kentucky bluegrass. We have Puccinia digitati coronata present in at least 3 states, North Dakota, Minnesota, and Wisconsin. Mostly POA species are affected locally of the grass species, some of them native. And common buckthorn is an easy host among other Rhamnus species, and likely facilitating the evolution of the pathogen, including important resistance genes in turf grasses potentially. Cereals are not affected significantly, and some grasses may host numerous crown rust pathogens or rust fungi in general, with the implication that potentially these are viable hosts if crown rust fungi are able to form interspecies hybrids, and potentially they're sites of genetic exchange between asexual or sexually compatible rust fungi. And I promise this one's really short. These are just 2 first reports. We're almost done. First reports of Puccinia glutamatis infecting sheep entirely, and of a rust fungus infecting lemongrass in Minnesota. This is a microcyclic rust fungus. A, we have the teliospores here, or the telia, and B, we have the teliospores. And actually, if anyone wants to remember another rust spore, This is a special kind of teliospore called a leptospore with really thin walls. They're not pigmented, and they germinate without overwintering, germinate rapidly. This is microcyclic, just teliospores and piscispores. It was first reported in North America in 1992, and it's been reported in states in the east and west of southern Wisconsin, and also in a few spots in the Pacific Northwest. It's been observed at 5 locations around the metro area by me in 2020, including on campus. And we provide evidence of continued clusters. And just like Puccinia coronata var coronata, this is an introduced pathogen from Eurasia on an introduced species, also from Eurasia, now reunited in their introduced range in North America. And now we have the first report of a rust, Puccinia species of lemongrass. We have the top and bottom view of a lemongrass leaf affected by the rust. B, we have a live micrograph of the Uraginius spores. Note the really thick walls, and also these long tubular cell separates the Uraginius spores. These are called periphyses. And C, we can see the texture of the Uraginius spore. It's what we call finely a kinulate. Eugene deserves credit for this observation. He saw it on lemongrass in 2022 in Mankato, Minnesota, on a community garden plot. Early reports of lemongrass in North America with other species, Puccinia nicholas in California, Florida, and Hawaii, where there's some lemongrass production. But in our case, the ITS sequence was 99% identical to this other species, Puccinia cistatii, which was not previously reported on lemongrass. It's also morphologically consistent. Our sample is morphologically consistent with this Puccinia cistatii, which is a known pathogen of lemongrass, although there's a lower ITS match according to publicly available, about 97%. And we just call it a Puccinia species for now. Still an interesting find, but really we need more phylogenetics work on these fungi with certified specimens to say anything for sure. So, in summary, we have 2 new crownless fungi in North America, part of a broader trend. We have PCC affecting highly invasive species, Glossy buckthorn would be canarygrass, of possible use as a biological control agent. And we also have Puccinia digitati coronata, affecting Kentucky bluegrass, other poa species, and common buckthorn. Crownless fungi are diverse and in need of more attention, so pay attention to them. And there are many new species around you all the time, so keep an eye out. So moving forward, there's lots of potential avenues of research to follow up on my work. Pablo has a new project through the Minnesota Invasive Terrestrial Pests and Plant Center to investigate the diversity of crownless fungi on common buckthorn and look at the relative abundance of these diverse fungi, both presently through surveys and also I hope historically through many varying specimens that people have collected over the years. We'll continue our work to evaluate the use of crownless fungi to control common and glossy buckthorns and try to associate various symptoms, which can be sometimes severe in common buckthorn, as you can see right, with various crownless fungi. Hopefully someday somebody follows up on this research. It won't be me, unfortunately, to assess the impact of PCC in field conditions for control of canarygrass. Maybe we can screen barley bagapodium with more crownless fungi. And ideally some more phylogenetic research, maybe with RNA-seq data or genomic data, to give us a little bit more definition in our understanding of the Puccinia series coronata. So thank you everybody for attending. Thank you very much to the Minnesota Invasive Terrestrial Pests and Plant Center for funding this research with funding provided by the Environmental and Natural Resources Trust Fund. So thank you to the generous residents of Minnesota. Thanks to everybody at the USDA Serial Disease Lab and the USDA in general, especially Les Sable, who's a co-author on a few of the publications, and Julia Mitchell, Sam Stockson, Josh, Samia, Melissa, Brenda, Kim, everybody, thank you so much. Everybody at the University of Minnesota, Nick Pajewski, Carl Miller, Eric Watkins, who have been co-authors on the publications, and to everyone else in my lab, GOT and Nisha. And really to everybody in the department, administrative staff, graduate students, researchers. This is a pretty pleasant place to be a graduate student. I'm very thankful. Thank you everyone, to my advisors and to my
Much needed. They don't have time for some questions.
I hope you have questions. Yes, definitely will do. Yes, please.
The lemongrass.
Mhm.
That's not
No, it's an annual grass. That's a really good question. I honestly, I don't know. It could be surviving in the plant, or maybe there's spores. So the woman who was farming it does take the lemongrass into her home every year, puts it in the cellar or something, and it just overwinters in her cellar. So maybe it's just like, yeah, hanging out in the plant tissue or in the soil. That would be my best guess. Kind of interestingly, we inoculated lemongrass in the lab and then forgot to water it, and the lemongrass just looked like it was dead, right? But then 3 weeks later, the plant came right back to life, and then a week later, there was rust. So it seems to be pretty hardy. Maybe it's living in the plant tissue. I think that's a good bet. Yeah.
For biological control purposes, how long, like, or how many like generations or years does it take for rust to eventually kill the host? Like,
so I've only seen crown rust kill very young seedlings of buckthorn. I mean, this is a woody plant, although there's some rust fungi that are able to kill woody plants. Crown rust fungi is certainly not one of them. It's once they're able to develop some amount of wood, it doesn't kill the seedlings anymore. I mean, I see some glossy buckthorn plants that have been just ravaged by crown rust year after year in some of these urban wetlands that are really infested with reed canary grass. And they look pretty bad and on the verge of death, honestly. So potentially with enough rust infection, you could just kill a plant by just knocking off its leaves every year. But in general, it's not a pathogen that kills plants. So, you know,
there won't be used biological control anytime soon in the greater Minnesota.
No, I don't think so. It would take a lot of work for sure. Yeah. Oh, and I'm sorry, I am not rephrasing the questions for the Zoom audience. I'm sorry, guys. I'll do.
Questions.
Oh boy.
When my daughter was young, we used to go out to we always thought the elders would bent on turtles.
Oh yeah. Yeah. So the question for the Zoom audience is: did I used to race painted turtles in Nisswa, where I'm from? And the answer is yes. That's our whole thing. Yeah. Mm-hmm. Mm-hmm. Yeah.
There's a future in biological control.
Maybe. So the question is, Nevin has reed canary grass, glossy buckthorn, and creeping Charlie in his yard, and he's mostly concerned about the creeping Charlie and hopes that the rust might kill it. And well, I know where you can find some inoculum. So really, yeah, I mean, go ahead. I mean, sure, it's probably coming. You can probably start through the telescopes right now. Yeah, I'll bring you a sample. You can try to inoculate your yard. Hmm. Mm-hmm. Yeah. Mm-hmm. Right. So the question is, yeah, how if we were to bring this to a natural setting and evaluate it in like in a natural setting, how would we evaluate the plants to see if the biological control is working? And yeah, I definitely want to consult some ecologists who have more experience doing like plant community ecology work. But certainly you can harvest above ground biomass, for example, and look at to what extent tree canarygrass is able to compete with like other plants that are there in like maybe a fungicide controlled area and a rusty area, right? So you could just, look at a biomass, look at amount of flowering, look at seed production, something like that, and compare it between treatments. I think that seems like a viable line of research. But yeah, I know that the ecologists probably have much more advanced methods, so we'd probably want to talk to them. Yeah, Matt.
So on the selection of marker genes,
Mm-hmm.
be more serious.
Mm-hmm.
So the one that got selected originally, of course, you had ITS. And the second round, you had or the other projects, you had ITS, beta 2, and 2 others, right?
Yes.
So the problem with those genes are that they're quite conserved. So when you go at the species level, it's fine, maybe. Unless it's very close to the species, then of course you should go within the species. Just further so looking forward towards the future, what would be your preferred strategy for sort of capturing both intra and interspecific variation for species?
So the question is, going forward, what kind of phylogenetics methods might we use to improve our understanding of the crown rust tree more broadly? And so I was working based on other people, the publicly available data, of course, and so other people have developed those markers. In the future, of course, we want some more resolution, right? So, and ideally not with herbarium specimens, because it's kind of hard to get good data sometimes from herbarium specimens. We want to collect live specimens of all these diverse crown rust fungi, which are really all over the world. And we'd probably want to do some, I don't know, RNA-seq or skin sequencing or GBS, just something to give us like full genome coverage to some extent, I think. If you have some ideas, feel free to share. Yeah.
You can also do the plants at the same time, right? So
Sure. Yeah.
The species of the plant that you're seeing.
Sure. Yeah. Right.
Because even that can be challenging, right? With some examples you can feel.
Yeah, for sure. And definitely, I think a major problem in rust phylogenetics work is that both the rust and the plants are often misidentified. And sequencing, of course, is a really good way to help with this. And previously sequencing was a little bit more expensive, a little bit more complicated. Now we're at the point where, it would be really nice to just have two sequences submitted together to confirm an association. Yeah.
Mm-hmm. How much do you know, or reading knowledge on how complex the population is in Devon itself of the pathogen population for each different pathogens?
Oh,
do you have much knowledge in terms of population size?
No, we really didn't do that work. I mean, certainly people have done that with oat crown rust, of course, but I don't think anyone's given that kind of attention to other crown rust fungi. I guess some with the perennial raggrass pathogen. There's some like RFLP work on that. Yes, Annie.
My first question in the chat.
Oh, good job, you guys. Oh, perfect.
Infected plants have poor seed development or seeds with lower germination.
That would be a really good follow-up study for sure, but we didn't test that just because we were limited in scope and time. Our plants didn't go to seed, so we weren't able to test that. Yes. Yeah, thank you.
Was it basically that it will post pictures of the wedding plans and that or did this
so the question is how I was able to use iNaturalist with this project. So this was partly successful just because of how you know, the pathogen is bright orange. It has sometimes really strong effects on the host, and the host is pretty widespread in populated areas, right? So we have those caveats. Like if you're looking for like a small like leaf spot fungus on some, you know, plant, there's not going to be people posting observations. But in this case, yeah, people saw it, they wondered what it was. Maybe they noticed that it was new and they posted it onto iNaturalist. So they just identified it as crown rust usually, or just identified it as all sorts of things actually, and as old crown rust. And I was able to just go on there and correct it, correct the identification to this organism. And then eventually other active users of the site caught on and were tagging me in posts and themselves identifying the rust. And I just, slid into their DMs and asked them for a sample. And many of them were very happy to comply. It was really as easy as that. Yes. Mm-hmm. Oh, sure. I should have explained this. It's really just like a social networking site where you just post pictures of like trees and mushrooms that you find. It's really very wholesome. It's actually gaining a lot of use, especially among the mycological community. So now with these like myco blitzes, where your groups of mycologists are locally trying to assess the fungal biodiversity, they're actually encouraging people to post every find on iNaturalist. And then later, when they're sequencing those samples, often with nanopore technology, actually, they are associating those iNaturalist observations and the serial numbers there with sequences in NCBI. So it's very easy to use. Don't let that scare you away. It's really easier than Facebook, honestly, at this point. But it's also very useful for scientists. And I think we're out of time here. So with that, I think we can close. Thank you very much.